Okay, ಕೇಳ್ತಾ ಇದೆಯಾ? Just tell me. ಕೇಳ್ತಾ ಇದೆಯಲ್ಲ?
ಹೌದು ಸರ್.
Okay. Uh,
ಹೌದು ಸರ್.
Yes, sir.
the only mutation in the germinal cell can pass on to their offspring because germinal cells are responsible for the production of male and female gametes. Suppose if the same mutation happens in the somatic cells, however good or bad that mutation may be, that is restricted to that individual only and will not be able to pass this character to the future generation, because all of you know the somatic cells will not take part in the reproduction, sexual reproduction. A point mutation is a change in one base in a gene. One nucleotide base changes means it brings the point mutation. Then what will happen? The effect of the point mutation may change. It can vary. Suppose if the mutation happens in a non-coding region of a DNA, absolutely it has no value. Most of the time, often no harm, often harmless. That such mutations will occur and it has no relevance to us also. Because the change in the nitrogen base sequence or the mutation in the nitrogen base sequence has happened in non-coding region of the DNA. Many times the mutation in the gene can be neutral because the redundancy in the genetic code. You know that there are same code, same genetic code for more than one. Sorry, for one amino acid, you have more number of genetic codes. So if this code fails, some other code will generate the same amino acid. So that's why if even if it happens, most of the time the mutations will be neutral because of the redundancy in the genetic code. Yeah, but sometimes the mutation which results in the change in the protein production, they are often harmful. Most of the time, I should say that these mutations, suppose if they are able to change the type of the protein that they produce, because proteins are the building blocks, which all of you know, they are often harmful only. And when such harmful mutations take place, generally they are not carried forward. But sometimes a change in the protein production can be beneficial also. But rarely, I should agree to the fact that results in the change in a protein production rarely becomes beneficial. And this may be carried forward. Even altering the gene number or position, which all of you know at the gross level when the chromosomal mutation takes place, either delete, addition, disruption, rearrange, which may which many loci are typically harmful. Even this. Changing in the rearrangement of the many loci on the chromosome. Most of the time they are typically harmful. But sometimes there's a duplication of small piece of DNA. If a small piece of DNA gets duplicated, what happens is it increases the genome size. The increase in the genome size is usually less harmful. I don't say that it is not harmful, it is less harmful compared to the rearrangement of many loci. And when they are less harmful, they may be retained. And when they are retained, a further mutation may happen to that, these duplicated genes. When they are exposed to further mutations, any new mutations, it may take up a new function. It may take up a new function. And it may be preferred also. Just to give an example, to detect the odor of anything, we have genes. This gene has duplicated number of times. The duplication has taken place in humans less and mice more. In human, we have more than almost 1,000 copies of the gene, duplicated genes. In mice, it is 1,300. Now, another fact I would like to draw your attention to the other fact also. Mutation rates are low in animals and plants. One mutation in every 1 lakh genes per generation. Remember, it's a very low because there is a proofreading mechanism during the protein synthesis. Even if there is a mutation happens, it will not allow that to happen. There is one mutation every 1 lakh genes per generation. Even mutation rates are very lower in prokaryotes, but in viruses they are very high. So that's why the mutation rate is in virus is very high. So that's why we are failing in finding vaccination for many of the diseases because they are mutation. Virus show the mutation, as you have seen in case of the AIDS virus. Till now, we are unable to bring any concrete vaccination because once you develop the vaccination, by the time you develop the vaccination, the virus mutates because rate of mutation is very high in case of the viruses. Then these mutations, if they have to pass on to the next generation, it is through sexual reproduction. Because not in the sexual reproduction, there's a reshuffling of the existing alleles to new combination. We have already studied the recombination of genes. There's a reshuffling, even swapping existing alleles into new combinations will happen. In fact, the organisms which are reproducing sexually, recombination of the allele is more important than the mutation because recombination is bound to bring variation because this is going to take part in the sexual reproduction. But when the gene mutation takes place, most of the time it may be redundant or it may be harmful or it need not be in the germ cell, it can be in the somatic cells also. But whereas the organism which reproduce sexually, there's a recombination during the crossing over process. That becomes more important to us than mutation and the genetic differences it produces make more adaptation possible in the hospital. So sexual reproduction has more role compared to the mutation. Now the question is how to test whether an evolution is occurring in a population.